Hi, I'm Dr. Kathryn Dao. I'm reporting on the ACR 2022 meeting for Room Now. And I have with us today a very special guest, Professor Eric Morand, who is the head of the School of Clinical Sciences and head of the Monash Health Rheumatology Unit. He also founded the Monash Lupus Clinic, and I'm so excited to have him with us today. Welcome, Eric.
It's great to be here, Kathryn. Thanks for taking the time to talk to me.
Absolutely. And, one of the abstracts that you're going to be presenting, it's abstract 2054. It's titled Towards a Novel Clinician-Reported Outcome Measures for Lupus, Outcomes of an International Consensus Process.
Yep.
So this is actually like a new treatment response measurement tool for lupus. Could you tell us a little bit about this and why it's so important?
Yeah, sure, Kathryn. So we all know that doing successful clinical trials for lupus has been very difficult ever since we started trying to do it. Part of the reason is the well-known clinical heterogeneity of lupus, the biological heterogeneity of lupus, issues with study design, steroid taper, patient variability, etc. But we also know that one of the measures is the outcome measures. So traditionally in lupus trials, what's been used is the SRI-4, which is based on the SLEDAI, and the main driver of improvement is a 4-point change in SLEDAI score, or the BICLA, which is based on similar tools, but just with different orientation, and you require an improvement in all BILAG domains, and they also have ways to capture no worsening. So they have worked. We've had belimumab approved using the SRI-4 originally. Not many people remember that the SRI-4 was actually derived from the belimumab phase 2 data. The original phase 2 trial for belimumab was negative, but the authors combed the data to try to find categories of patients who looked like responders and measures of improvement that looked like a measure that worked, and that became the SRI-4, and it's worked for every belimumab trial since then. It's worked for very few other trials, however, and famously the TULIP 1 and 2 trials were discordant in their results for SRI-4, although concordant in results for the BICLA measure. And these were identical phase 3 trials using the same compound, anifrolumab. We're lucky that anifrolumab ended up getting approved because of the totality of evidence, but actually it was a narrow thing because that primary outcome measure, SRI-4, was negative in the TULIP 1 phase 3 trial. So while there remain many reasons for this, we're quite sure that these tools actually are part of the problem. So the reason for that is that the SLEDAI and the BILAG both go back to about 1990 or 92 when they were first described. And although they've had improvement since then, and although they were derived from a very hardworking consensus process, they were not designed actually to be used in clinical trials. They were used as for use in cohort studies to measure disease activity, over time. And so on. They weren't specifically designed to measure response to therapy. So they've been hijacked into this role to measure response to therapy, for which they were not designed. They have performed, the results are mixed, and we think that the time is right for a dedicated effort to make a response measure that's just designed for one thing, which is measuring treatment effect in a clinical trial. Because we still need more treatments for our lupus patients, as you know, Kathryn. We've got a long way to go to get better outcomes from our patients.
Absolutely.
We can't afford to keep having trials that fail due to basically to a problem that we caused, because we're the ones who make up this measure. The biology of the lupus is the biology of lupus, and the molecular target is the molecular target. But the measures are made up by us, so it's up to us to get that right.
Yeah, I've been involved in quite a few of the clinical lupus trials, and it was so frustrating, because you can see a treatment response, and these patients, though, are on such high background medicine, so then you don't see the treatment effect. So how exactly are you going to derive all these definitions of what you would deem appropriate?
Yeah. Thanks, Kathryn. So what we decided to do, when we began, we started talking to colleagues and also to industry partners because it's our patients and us and the pharma companies that need this better tool. So it's sort of all of us who need it, and all of us have to work together to get it. And we started thinking of this chiefly as a scientific project. And my own feeling was that this would be potentially quite doable if we borrowed from a very good measure like the ACR 2050-70, which has stood the test of time and worked great, given us fantastic medicines to use in our RA patients, and is based on using continuous variables like swollen joint count or CRP, measuring percentage change from a baseline, and the baseline is that patient's baseline, and every patient has their own baseline. So we don't do any of those things when we do lupus clinical trials. We don't use individual baseline, we don't use continuous variables, and we don't use percentage change. So we started to wonder, what if we took that approach and looked at things that we do actually measure in lupus, like a joint count or SED rate, or platelet count, or proteinuria. These are continuous variables. They would be able to be measured in a similar way to what we've just described for the ACR 2050-70. Is that something that we could do? So that was our starting point. But then we realized that actually to do it right, we needed to actually throw away our starting point ideas and start with a blank page. And so that's what we've done. And in this abstract, we describe a process wherein we've gathered a consortium of clinical academic lupus experts, experts from 10 pharma companies involved in lupus drug development, leadership from the Lupus Foundation of America and Lupus Europe, representing patients, a very robust governance committee, including a patient advisory panel, scientific advisory board, steering committee. And that group of people have sat around designing a method to come up with a new measure for lupus clinical trials. So even though we've started with some ideas, we've actually set them aside and focused our attention instead in trying to have a robust scientific methodology to go from a blank page to a measure that works using the best possible evidence, expertise, and Delphi consensus methodology, nominal consensus techniques, and just try to be really methodologically robust.
And I noticed that you've had some pretty heavy hitters in the world of lupus. How hard was it to get all these people to come together? Because you have people from North America, Latin America, Asia, Australia. So the time zones are different. The opinions are different. They're very strong-willed. So how was that like?
Actually, it's great. We found that there's been a strong agreement on the need. And there's been a kind of a rallying behind the idea of being just really agnostic to what the outcome looks like. It's not my measure or your measure. No one's bringing their measure. We're going to use a process, a collective process to get to the point we need to get to. I need to point out that we've also have regulatory experts, and that turns out to be a hugely important part of the process. And I'll talk about that in a second if you want me to. So actually bringing people together, I think everybody wants this. And if you lay out a robust methodology with strong governance, and it's really a partnership, people are interested to spend their time. In terms of the time zone, I'm from Australia, and we're used to having to get up in the middle of the night to meet other people's time zones. So that's what we do. It's definitely challenging to have Europe, Asia, North America, South America, but actually we wanted to have global representation from lupus clinicians, global representation from companies involved in developing medicines, and now we have global representation on our patient advisory panel as well. Underrepresented non-European ancestry people is an issue in clinical trials. It's an issue in research, and we just wanted to cover that base as well.
That's what I thought was the very strong point, is the representation from the diverse background. And you had mentioned regulatory experts. Was the FDA part of this, and what other regulatory governance was involved?
Well, what we've realized in the end is that we could do the best science in the world and come up with some kind of amazing, highly effective discriminatory measure. But if regulators like the FDA don't like it, we wasted our time. Because in the end, for a company to have a product approved, they need to do a trial with a measure that the FDA approves before they commence the trial. So right now, the FDA approves the SRI and BICLA for lupus trials. Notwithstanding the issues that are there, we'll need to provide high quality scientific proof that we've got something better. And we need the FDA and the European agency, the EMA, and all the other national regulatory agencies to get on board. So we've engaged some regulatory experts, some ex-FDA people actually are on our steering committee. They're guiding us about how to get it right. Even the wording is special for regulatory documentation. I've learned phrases that I never used before, like concept of interest and context of use. And in our abstract, we define our methodology that we've yielded definitions for those two things for our process. And what we're about to do, actually right after I get home from the ACR, we're going to be formally writing to the FDA with an outline of our proposal. They actually have a process to look at new measures. They have an evaluation process that we're going to engage in. And what we hope to do is to get the FDA not across the table, but around the table. So we actually work with them. So we're about to reach out to them. And if anyone is watching from the FDA, please contact me. We need to do that first with the biggest agencies that are so influential in having drug approvals, that Europe and the US. But in the end, that'll need to be replicated around the world. But I guess we can't do everything at once.
Now, I know that you're using the PICO-C framework to develop this tool. Mm-hmm. And the consensus is actually pretty high across the board. But the lowest one was actually the patient item. There's 64 items. Mm-hmm. And the consensus is, I believe, 81%, whereas all the others were above 90% or even 100%. Yep. Will you talk about that? What happened?
Yeah. Well, so we used the PICO-C framework for one specific task, which was to define this conceptual definition. What we understand now, in order to meet the regulatory requirements, is we start with a conceptual definition of what we're trying to do. And after that, we lay upon the kind of methodological, operational, mathematical components, which won't be done with PICO-C. That'll be done with other techniques. So we had a preset consensus threshold of 70% to be acceptable. So 80% was fine. That we had a kind of a priori gate of 70% consensus to move forward. So we weren't worried by having 80%. You know, these consensus processes, humans are involved. And at the longer we did the P first, and we did them in order. And I think we got to know each other better. And we kind of concepts that we came up with, we did our job better, and we got higher levels of consensus as we went through the process. So I think that's not unusual for this kind of approach, and we think those numbers are fine.
Part of me felt like you were so tired by the end. You're like, oh, what the heck?
Well, I hope not. And we have to try to resist that sort of thing.
Right. Right.
We have to work really hard to resist that, because this is, why hasn't this been done before? Because it's hard, actually. This is a lot of work. The process that we've just completed is a survey of physicians and patients for the domains that they consider important in their disease, in the disease lupus. Not how to measure it or what change looks like, but just what is important. And that was an uninformed survey. We've got a long list of items when we're about to do a massive series of literature reviews to find evidence for whether or not there is evidence supporting the importance of those things in terms of what the FDA refers to as how a patient feels, functions, or survives. And the language at the FDA is extremely patient-centric. The movement is really strongly away from what does the doctor think to how does the patient feel. And so we have strong engagement with patient organizations and directly with patients on our patient advisory panel. And there are patient members on each of our steering committees as well. So we will be driving strongly towards this idea of the patient at the center of the story. And I think that's going to give us a measure that's really different to what we've done before. Well, that's okay, because what we've been doing before hasn't always been working so well.
That's so important. And that's why I really love the fact that you all have gotten together to formulate this tool. Because I believe in the abstract, it says, we're not just going to be measuring disease activity, but we're measuring treatment response. And this is patient-centered.
Right, exactly. So we are not trying to design a new disease activity score. Not at all. A side product might be a disease activity score, but that is not our goal. Our goal is to measure delta in response to therapy in the context of a placebo-controlled trial. So that's our single focus. Now, it might be possible that such a tool could also be used in everyday clinical practice, or it might be possible that such a tool would find a use in cohort studies of disease activity, but we don't want to kind of pollute our agenda with those possibilities, because we might get distracted from doing the one thing that we need to do. And, it's science, so we don't know the result yet. We don't know whether we're going to succeed, but we don't want to fail for lack of trying.
Right. How will you validate the tool?
Yeah. So the abstract describes what we call aim 1 of our project, and it has 3 aims. Aim 1 is to basically come up with a novel measure. We'll be reporting further outcomes of our consensus process over the next year at the next upcoming meetings. Once we have the draft measure, we'll then be working with our industry partners to validate it in trial data sets, which we don't have access to, but which the companies have committed to go away, do the work inside their shop on their data, come back with the results and share those results with the whole group. And I must say there's been fantastic, very open and transparent collaboration between these companies who, commercially speaking, are competitors, but they're extremely collaborative in regard to this, including in their commitment to use data to help address this. So aim 2 of the project will be to use existing trial data sets and new clinical trials, because companies are starting new trials all the time. So as the first trial to kick off once the instrument is in draft stage, will include this instrument as a exploratory outcome measure. So we'll get real-time data as new trials go forward, as well as look-back data using existing trial data sets. And then the third, and I think very important aim, is to validate this measure in terms of long-term patient outcomes. Because actually for SRI and BICLA, there's very little validation that attaining an SRI or BICLA response actually changes your outcome in terms of damage accrual, health-related quality of life, mortality, and so on. I mean, there is some evidence now in the health-related quality of life that a BICLA responder, for example, has improvements in health-related quality of life, but there's not vast amounts of evidence in that space. There's just a bit. So we propose to reach out to long-term registries to deploy this tool and track patients' response in the real world and see whether it correlates with those outcomes that are so important, like damage and mortality. So those are the 3 parts. It's going to take a while.
Well, I'm definitely looking forward to it, and looking forward to hopefully incorporate this tool in future clinical trials that we'll be involved in. So I want to thank you so much for your time, for allowing me to pick your brain on this, and congratulations again on putting together such an incredible team to produce such an important tool. This is Dr. Kathryn Dao reporting for Room Now. Please follow me on Twitter at KDao2011. Have a good day.
Bye-bye.